Good afternoon, my teachers and my classmates. The topic of today's seminar is T-cell lead peroxidation induces ferroptosis and prevents immunity to infection. So in this title, you can see that T-cell can undergo lead peroxidation. And when lipid peroxidation occurs in T-cells, ferroptosis will be induced and immunity will be compromised to infection. So first, let's introduce the development of T cells. We can see that T cell comes from hematopoietic stem cell in the bone marrow, and it will further differentiate into common lymphoid progenitor. In the next step, it will differentiate into cell precursor, and it will enter into the thymus. And in this process, the T cells can be formed based on multiple stages. So we can see in the next slide. The multiple stages include three kinds of cells, including double negative T cells, double positive, and single positive T cells. So in the second stage, which means double positive into single positive T cells, the cell fate will be determined based on the molecules that are bound on the macrophage or dendritic cells. Next. Also, you can see that ROS is a very important factor in the pathways of T cells activation, expansion, and effector function. And the ROS can come from NADPH oxidases and electron transport chain from the mitochondria. So here's the previous results. Now, there's a synergistic relationship between selenium and vitamin E to inhibit peroxidation of lipids. And we can see that selenium is incorporated into GPS4. So we can know that GPS4 is also involved in the lipid peroxidation pathway of the scavenging network. Okay, so we can know that GPS4 is very important for ROS scavenger. So how about its role in the immune system? Next, the main objectives of this paper is to investigate the GPS role in T cell responses, including thymine development, peripheral T cell homeostasis after development, and the activation of T cells after infections by either virus or parasite. So first, the other want to investigate the main role of GPS4 function in the T cell mediated immunity. So the knockout mice will be generated through the Krylospe system, because in the previous paper, it has been shown that when the mice were homozygously knocked out in GPS4, the mice will die at the embryonal stage. So to prevent this lethality problem, Krylospe system is employed because Krylospe system is very tissue specific. These techniques will be explained further. Okay, and next, cells will be isolated from the mice using these two techniques, and these two techniques will further be explained. And next, the cells will be quantified based on the specific cell surface proteins and to know which kind of cells have the highest amount or lowest amount compared to wild type. So first, let's introduce the Krylospe system. We can see that two strains of mice will be generated. One is to express Kry recombinase, and the other one is to express the fluxed GPS4, which means the GPS4 is sandwiched between two box peak set. And we can know that when these two strains of mice are crossed, the next generation will not have the gene of GPS4. Next, we can know that the T cells can be isolated from these two techniques. The first is FACS-A area L3U. This process is based on the fluorescence molecule bound to specific kinds of cells, and this fluorescence can be detected by laser and then be charged with a specific charge, either negative or positive charge, so can be separated and then after this first process of separation, it will go through MACS microbead separation, and it will based on the magnetic separation. Because the microbead is covalent bound with antibody, so this antibody can bind to specific kinds of cell surface protein. So only those are bound with magnetic microbeads will be retained when the tube is put into the magnetic field. And next, the cells with microbeads can be eluted when the magnetic field is removed. So after the isolation of T cells, the cells will be quantified using a specialized kind of flow cytometry called fluorescence activated cell sorting analysis. And it's very similar to the first technique we had explained for the cell isolation. The cells will be bound to specific molecule and go through a laser to take on specific charge, and it can be separated. And after a separation, we can know specific amounts relating to specific kinds of cell surface protein the cells bears. So we can know that in the first experiment, in the first results, the flow cytometry diagram is here. And there are different numbers. To compare numbers, the same regions of numbers will be compared because the same regions means the same kinds of cells. So we can know that these regions have very similar numbers. There's no significant difference in numbers when we do the bar chart and make the statistical comparisons. And we can know that there's no significant difference, which means GPX4 is not involved in the thymic T cell development. Next, we can see that after the development, fewer CD8 plus T cells will be in the spleen peripheral lymph nodes in the knockout mice, which means the GPS4 is very essential for homeostasis of CD8 plus T cells in the periphery, but also CD4 plus, but in the results we cannot see that. Okay, so the next method, the method change, the results can be confirmed because this result is quite contrary to the expected results because these two cells involved in the same stage. So it's very strange that only CD8 plus T cells will be affected but not CD4 plus T cells. Next, we can see that T regulatory T cells homeostasis will not be affected by the lacking of GPS4. And next, we can also see that the activated form of memory T cells will not be affected when the GPS4 is absent. And in these two figures, we can see that there are different kinds of cell surface protein. It is very specific for the T memory cells or T regulatory cells. It can be detected. So to know that, maybe CD4 plus T cells also is involved in the system, but it cannot be detected the difference due to some barriers. To change this method, the author used the cells to transfer into the wild type instead of just analyze the cells from the original knockout mice. So to do this, the original cells from knockout mice will be mixed with the wild type cells with equal ratio, and it will be transferred into a healthy wild type male mice. So we can know that these two cells will compete with each other, and when the expansion will go greater, which means this kind of cells will have the greatest amount. However, if the cells both have low amount, that means the mutant type cells has already disrupted in its homeostasis. So we can see that in the next figure. There's no difference in the double positive and single positive thymocytes, which is very consistent with the previous results. But we can see that in this figure, both CD4 plus and CD8 plus homeostasis will be greatly affected by the absence of TPS4. So the absence of TPS4 did not interfere with normal thymic T cell development, but it will affect the homeostasis of CD4 plus and CD8 plus effector T cells in some parts of the secondary immune system. And also, the absence of TPS4 did not affect both regulatory T cells and memory T cells survival. So after we know that effector T cell can be affected when the TPS4 is absent, we want to know that after the activation induced by the infection, will this stage also need TPS4? So to know this question, to know the answer to this question, the mice will be first infected with specific peptide segment from the virus. Why choose specific peptide segment? Because this segment is already known, so we can detect specific cells that are activated due to this infection using NAC class 1 and class 2 molecules. And again, these mice will be analyzed after the mice has been processed. So you can see in this figure, both CD8 plus and CD4 plus T cells were undetectable in the spleen when the TPS4 is knocked out, and it cannot be detected by NAC class 1 and NAC class 2 tetramers. In this figure, we can see that the knockout mice have significantly lower number compared to a wild type, either in CD4 plus T cells or CD8 plus T cells, is a great significance. Next, the author changed the infection from virus to parasites and do the same experiment. And also, we can see that the CD8 plus and CD4 plus T cells are affected if the TPS4 is absent. But in this conclusion, the author only focused on CD4 plus T cells. That's because the parasitic infection, mainly CD4 plus T cells were involved, not the CD8 plus 1. So in summary, TPS4 deficient T cells can immediately collapse after either viral or parasitic infection. And in the parasitic infection, especially is the T helper cells that dies when the TPS4 is absent, which makes the immunity compromised to infection. So next, the other one to know, how are these cells undergoing the death pathway? Maybe there are some parameters we can see to know whether or not these cells undergo this cell death. So first, the other one to investigate the association between this kind of cell death and lipid peroxidation. So lipid peroxide will be detected by C1 body staining. After the staining, we can see the levels of lipid ROAs, but also to confirm the association, a rescue assay will be performed using the epsilon. Because epsilon is a GPS-like agent. It is an antioxidant, so it can scavenging ROAs, making cell death less likely to happen. So we can see that, Body staining, because body is originally in the reduced form. After it encounters lipid peroxides, it will be oxidized and becomes the green color. So this shift can be detected by spectrometer, and the absorbance can represent the levels of lipid peroxidation. So in these results, we can see that in the first time frame, 0.5 hours, there has been already the peak shift compared to wild type, which means cell death happens greatly when the lipid peroxides are greater. And you can see that as time goes by, only the difference will go higher and higher. So we can see in the rescue assay, when epsilon is added, as the amounts are greater, the cell viability will be greater, and in the highest amount of upselling, 30 micromolar, there has been no significant decrease compared to the wild type, which means RS is greatly associated with this kind of cell death. And next, the other one to know the exact cell death pathways about the lacking of GPS4 in the T effector cells. So to know this question, to know the answer to this question, the series of established inhibitors specific for a cell death pathway will be tested. When specific inhibitors added can prevent cell death, that means this cell death undergo these specific pathways. So in the result, you can see that only the inhibitor specific for the pathway, ferroptosis, can inhibit cell death. The cell viability will go up greatly compared to other inhibitors. So it can further confirm that the cell death will undergo ferroptosis. The cell death is carried out by ferroptosis. And we can know that the science-based cell death was due to uncontrolled levels of their peroxidation. So we already know that their peroxidation is a key factor for the cell death. So we want to know some source of RS from this cell death. So to know this, the author make a hypothesis. Maybe ALOS15 is very likely to be involved in the source of RS, in the generation of RS, because ALOS15 has been observed in the previous research that it can cause the mice to become dead. Fibroblasts, fibroblasts will become inactive, it will die because of the overexpression of ALOS15. Okay, so ALOS15 is a apoptosis inducing factor. So we can know that it's very fatal. So maybe it will be involved in the ferroptosis too. So to know this answer to the question, the ALOS15 knockout mice will be crossed with the GPS4 knockout mice. So the offspring will have a double knockout mice, and the mice will be infected with the virus, and the cells can be analyzed after the mice have been processed well. So in the results, we can see that there's no significant difference between single and double knockout mice. We can see that in CD8 plus T cells and CD4 plus T cells, the double knockout mice compared to the single knockout mice in GPS4, there was no significant difference. The bar has similarly height of bar. The bar is very similarly high. So it means that maybe ALOS15 is not involved in the generation of RS to cause fibroblastosis. And maybe some rules will be evolved, including 5-lipoxygenase or other non-enzymatic oxidation or cellular RS mechanisms. And based on the previous observation that alpha tocopherol can inhibit cell death, so there comes a hypothesis. Injection of vitamin E can prevent GPS4 deficient T cells from undergoing ferroptosis. So to test this hypothesis, the mice will be fed with different amounts of vitamin E before the infection and cell analysis. So in these results, we can see that when the mice are fed with high dosage of vitamin E, there will be greater cell viability. The cell numbers are greater, and in the CD4 plus T cells, we have already seen that there will be no significant difference compared to a wild type. So the dietary supplementation of vitamin E can restore peripheral CD8 plus T cell, but not just only CD8 plus, but also CD4 plus. Yeah, we can see in this figure. Yeah, it's very obvious. Yeah, although CD8 plus T cell, there's still significant difference, but the levels goes down. So to know, maybe GPS4 will also evolve in secondary T cell response. A different method was employed, a modified version of curiosity system, because we want to only test the absence of GPS4 influence on the secondary T cell response. The GPS4 will be knocked out after the first infection, so to only see the second infection. So to do this experiment, the mice will express both query recombinase and flux GPS4, but this query recombinase will be bound to HSP90, so it will not be entered into the nucleus when tamoxifen is not administered. Only when the tamoxifen is administered will the HSP90 detach from the protein complex and make the query recombinase enter the nucleus to knock out the GPS4. So we can see that in the results, the difference is very small. Yeah, there's no significant difference. Yeah, it's very comparable between the knockout GPS4 mice and wild-type mice in relation with the secondary T cell response. So we can know that vitamin is a potently antioxidant that can potentially compensate for the lack of GPS4. Also, GPS4 is essential for primary but not a secondary T cell response. In conclusion, the absence of GPS4 disrupted the homeostasis of T effector cells in the secondary immune system, and also it can cause effector T cells to undergo apoptosis after infection-driven activation. And the last, dietary supplementation of high-dose vitamin E could reverse the effects of GPS4 deficiency. So it may be a very great antidote for the genetic disorder or any environmental caused disease. So here's the end of the slide. Do you have any questions?